Welcome to this video lecture. The topic for this video lecture is differential contrast interference microscope, one of the techniques in microscopy which is very useful for the 3D observation and image. So let us see the learning objectives. The first one is to describe the principle and working of differential interference contrast microscope, DCIM, and then we shall state and focus on the applications of differential interference contrast microscope. As you can see, it's an image taken from the book Prescott, where it's a micrograph of the protozoan amoeba proteus, and the 3D image, it contains considerable detail and it is artificially colored. It is 160 times magnified. Let's proceed with the principle of differential interference contrast microscope. It is very much similar to the phase contrast microscope. So it's a request to all the viewers and my dear students, if you have not yet checked and observed and heard about the video, and if you have not yet checked the video for phase contrast, so it's a request first to study the phase contrast microscope in detail, and then if you would come to DCIM, it would be better for you to understand. So let's proceed further. So DCIM is very much similar to phase contrast microscope in that it creates an image by detecting the differences in the refractive indices and thickness. So as we had discussed for the phase contrast, here also it is detecting the difference in the refractive indices and thickness. So what happens is two beams of plane polarized light at right angles. So please note there is a role of plane polarized light. So I believe you know the basic physics. We had studied this in the junior college also, what is meant by a polarized light and the plane of the light. So in this technique of microscopy, there are two beams of plane polarized light. They are at right angles. So they are at a right angle, means at 90-degree angle to each other, which could be generated by use of prism. So in one design, the object beam will pass through the specimen, while the reference beam will pass through a clear area. So because you're mounting a specimen on the slide, so some of the part and region of the slide will have the specimen and some will be clear area. So in one design, the object beam will pass through the specimen, while the reference beam will pass through a clear area. So there are two beams of light. So once it is passed through the specimen, the two beams are combined and they will interfere with each other to form an image. So what happens is, in this you can observe a live specimen which is not stained, and it will appear brightly colored and you would have a 3D image, a three-dimensional image. So what are the applications of DCIM, differential interference contrast, DICM to be more specific? So structures could be cell wall, endospore, granule, vacuoles, and eukaryotic nuclei. They are clearly visible. So for this particular topic, the reference is Microbiology by Prescott, or 5th edition. So you can refer to this book for your further studies. So thank you for listening to this video. Keep exploring the world of mini wonders.